Thank you. And first, I'd just like to start off by thanking the organisers for giving me the opportunity to present here today. As a researcher working at the bench, I've really enjoyed listening more to the clinical side. Sometimes I feel a bit disconnected from that, so I think these kind of meetings are really important. So I'm based up at the MRC Centre for Medical Mycology up at the University of Aberdeen. And today I'm going to talk to you about my research, which is focused on developing fully human antibodies for potential use as diagnostics and therapeutics, with an initial focus on Candida infections. Oh, okay. Okay, so as many of you obviously will be aware, invasive fungal infections have a significantly high mortality rate. This is higher than the number of deaths caused by malaria. 90% of the life-threatening fungal infections are caused by species from the 4 major human fungal pathogens: Candida, Aspergillus, Cryptococcus, and Pneumocystis. Now, within these, Candida species are now the leading cause of healthcare-associated bloodstream infection in some US centers. So, talking about candidemia and invasive candidiasis, so who's at risk from these infections? Well, it's patients with severely weakened immune systems, so those in the ICU setting, patients undergoing surgery, or those on chemotherapy for cancer treatment, and also transplant patients as well on immunosuppressant drugs. Now, despite treatment, up to 40% of patients will die from these infections. So why is this number so high? And as a community, what can we do to address these issues? Well, we've already heard a lot about the issues in terms of diagnosing these infections. And looking specifically in terms of Candida infections, we need to develop more rapid and discriminative diagnostic tests, which are point-of-care use. We also need to develop new therapeutic strategies, new classes of drugs with new mechanisms of action. And these ideally will be safer than the current ones we have available, and as we've just heard about, effective against resistant organisms, or have the potential to slow down this resistance development. Finally, we need to get vaccines to fungal infections into the clinic. Currently, there are no vaccines in the clinic for any fungal infection. Now, over the last few decades, there have been huge advances made in terms of antibody engineering and monoclonal antibody technology. And there are currently over 63 antibody products on the market in other disease areas, primarily cancer and autoimmune disease. However, despite their huge impact in these areas, they've yet to be fully exploited in the fungal setting. And this is really what prompted our vision in Aberdeen to generate fully human monoclonal antibodies for potential use as fungal diagnostics and therapeutics, using a novel single B-cell technology and Candida albicans as the model organism. So I'm just going to run through the technology very briefly. So we took class-switch memory B cells from patients who had recovered from these Candida infections and activated them to secrete antibody, which was then screened against defined protein targets or crude whole cell Candida preparations. The positive hits were then taken forward, and the variable domain heavy and light chain genes cloned directly from single B cells and then expressed in mammalian cells and purified. And the outcome of this was that we generated a panel of 18 fully human IgG1 monoclonal antibodies directed towards Candida. So 5 of these bind to HYR1, which is a hyphally regulated protein expressed on Candida albicans hyphae. And the remaining 13 were isolated against these crude whole cell preparations. So we know that they're binding specifically to the Candida cell surface, but we haven't identified the exact target for these. Okay, so why are we so excited about the potential that these may have? Well, as I've mentioned already, there is a huge number of these already in the clinic, and our human IgG1 format is the most common isotype of these in the clinic. These are targeted to the fungal cell wall, a structure which is not present in human cells, and therefore there's the potential that there's less of a chance of getting any off-target toxicity in the patient. Because these were generated in the human in response to an infection, theoretically, they are more specific to human markers of infection. This class of drug has a half-life which is around 20 times longer than current antifungal drugs. And these IP-protected antibodies we have shown are effective against resistant organisms. So now I'm just going to talk through a bit of the initial characterization we've done with this panel of antibodies. Okay, so first of all, looking at the HYR1 antibodies. So HYR1 was originally identified in Aberdeen, and it's expressed on the Candida albicans hyphal cells. And actually, the reason that we chose this target is because there was evidence in the literature that a fragment of this protein showed protection as a vaccine candidate in a mouse model of disseminated candidiasis. So we screened against the recombinant protein antigen. So the first thing to check was, do these antibodies bind to HYR1 expressed on the native cell? And as you can see here, with the immunofluorescent, we see only binding to the hyphal part of the cell. And then consistent with the literature, we see only binding to Candida albicans hyphae. We don't see any binding to any other species of Candida. And indeed, we don't see any binding to Candida albicans in its yeast form either. In terms of the anti-whole cell antibodies, so these could be split down into 7 different clusters dependent on their VHCDR3 amino acid sequence. And so here is shown is just one representative from each of these different clusters. And these 4 here, as you can see on the top with the immunofluorescent microscopy, these bind to targets which are abundantly expressed on both the yeast and hyphal parts of the cell. And this is confirmed through TEM imaging, where we see nice localization of the antibodies at the yeast in this top panel, and then the hyphal cells as well. We have 2 clusters represented by 126 and 131, which bind predominantly just to the hyphal part of the cell. We do see some binding to yeast, but this is very minimal. So these are predominantly hyphal binders. And then we have antibody 127, which is really interesting. It shows punctate binding to the mother yeast cell, but then also binding solely to the hyphal tip. And we also observed this with the TEMs as well. So that was all done looking at Candida albicans. The next thing was to see, do these bind to other species of Candida? So as I mentioned previously, for the HYR1 antibodies, we found those to be Candida albicans specific, at least against the species that we've tested. For the anti-whole cell antibodies we have seen more pan-Candida activity. So we see higher binding to those species which are more closely related to albicans, and such as Dublinensis, Tropicalis, and little to no binding to, for example, Glabrata and Crucii. And interestingly, we see nice binding for some of these against Candida auris, which could be very important in the future. We didn't see any binding to Aspergillus, Cryptococcus, or Pneumocystis species that we tested, so at the moment we are defining these as Candida specific. Okay, so I'm just going to take one antibody out of this panel and go into a bit more detail about its target. So this antibody binds to a target expressed abundantly on the yeast and hyphal cells. And through the TEM localization, we hypothesized that this target was likely to be a proteoglycan epitope with a core N-mannan structure due to its localization between the interface between the outer and inner cell wall here. And this was backed up by polysaccharide screening that we did. So on the left, you can see that we screened against a number of different fungal, bacterial, and plant polysaccharides and included the Candida N-mannan. And we see a nice peak here for Candida albicans N-mannan, and then no cross-reactivity to any of these other polysaccharides tested. We then went on to look at mammalian and glycans and saw no cross-reactivity to any of these, which is obviously very important if this was to go forward as a drug. And this antibody is Candida specific. So this one binds really nicely to albicans and to these species here, but has little or no binding to Crucei and glabrata. So after doing all this binding analysis, we then wanted to see, does this have any functional effect? And then, so my colleague Adelia introduced me to this quote, which I really like because I think it sums up our approach at the moment and also what I'm going to show you over the next few slides. So stimulate the phagocytes, drugs are a delusion. So on the next few slides, hopefully, my videos will be working. So I'm going to show you movies of Candida albicans being phagocytosed by macrophages in the presence of a control antibody or one of our antibodies. Oh, that was me. Oh, no. Right, I'm going to try this again. Okay, so on the left-hand side, we have the control antibody, and on the right is our anti-Candida antibody. And I hope you can see the profound difference here between the two. On the right-hand side, the macrophages move a lot more quickly and directly to the Candida cells and engulf them a lot faster, ultimately leading to earlier clearance of fungal cells from the assay. And interestingly, we see exactly the same thing with Candida auris, which is really exciting, especially given the huge problems potentially that we might get with this multi-drug resistant organism and looking for new options for treatments. And this work that we did in vitro translated to protection in a 3-day mouse model of systemic Candida albicans infection. And this was determined by kidney fungal burden reduction and a reduction in overall disease outcome score, which also takes into consideration the weight change of the animal as well as the fungal burden. And this is currently something that we're looking to add to this data set to strengthen this proof of concept. I haven't really touched on this today, but there is also huge potential to use these as diagnostics. And we've been working with an SME partner to develop a lateral flow device for this. And as you can see here, we've taken the nice high specificity binding that we see down the microscope and converted that to a red line on a lateral flow test. And this is detecting Candida antigen in just a Candida culture sample. So we've yet to test this with clinical samples. That will be the next step. But we're encouraged that we've also tested this against looking at Candida supernatant as well, and we see binding. So yes, I guess you never know until you test the clinical samples, but we're looking forward to doing that. And just finally, so I'm currently funded by Scottish Enterprise through a Royal Society of Edinburgh Enterprise Fellowship. And that is to commercialise this research from the MRC Centre for Medical Mycology. And this funding over the next 18 months will hopefully allow us to get the key proof of concept animal data that we need to secure investment, ultimately spinning out the company Microbiologics probably early 2020, focused on developing these human antibodies as potential new treatments for Candida infection. So just to summarize, new fungal diagnostics therapeutic strategies are urgently required. I don't think I need to really say that here. Human antibodies, I think, provide a really interesting and new approach to this area. And it's really encouraging to see that we have a whole kind of session on antibodies in the field at the moment. So that's really exciting. Through our work, we've generated 18 anti-Candida antibodies through this single B-cell technology and shown opsonizing activity in vitro and protection in vivo. There's huge potential here as well to develop a future companion diagnostic with our goal to commercialize this and really get this up and running as a company called Mycobiologics. And finally, there's a lot of people to thank. Most importantly, perhaps Neil Gow, who I'm sure a lot of you will know. He was my supervisor when I started off this project as a PhD student and is still heavily involved in the project. And then, yes, there's a lot of other people. I probably don't have time to go through them all, but yes, they're all up here. But then if I could just abuse my time on the stage a bit longer, I just want to put in and ask. I'm really interested to hear from people who are working with patients and, who are trying to diagnose or, trying to treat these patients with these infections to get a better understanding of, what you would like to see from a new therapeutic or diagnostic, and just to understand the process a bit better. So please come up and talk to me in the breaks or this evening. I'd really appreciate it. Thank you.